if we want to understand how CO2 in the ocean might affect carbon isotope fractionation, we've got lots of things to constrain, right? Well, one thing that we can constrain is surface area to volume. And we can do that by targeting a couple species in the ocean. This is one of them. This is Emiliani huxleyi. These are those calcium carbonate plates that they make. So this is just a scanning electron microscope image. What is unique about Emiliani huxleyi and a related species, Gephyrocaps oceanica, is not that they produce these calcium carbonate plates, but they produce these weird compounds, these things called alkanones. They're a homologous series of C37, C38, C39 ditri and tetraunsaturated methyl and ethyl ketones. So they have a number of unsaturations here, double bonds in these ketones. They are unique, and they make a lot of them. They're probably storage products, but the weight percent of these compounds is surprisingly high, like 10 to 12%, maybe even higher in some. But they produce lots of them, and they're unique to these two species. So with that, if we can analyze the carbon isotopic composition of these compounds, we can begin to constrain at least one of the factors that controls carbon isotope fractionation in marine microalgae, surface area to volume. So there's been a concerted effort to use these alkanones for that, or use the carbon isotopic composition of these compounds for that. We're also lucky that these are the most abundant of the coccolithophores in the ocean. They're very cosmopolitan. They occur in all but the polar oceans. Okay, so if we look at a laundry list of the factors controlling carbon isotope fractionation, certainly we have concentration of CO2 and growth rate, cell geometry, the isotope effect associated with Rubisco and all carbon fixing enzymes. If we look at different forms of Rubisco, they're 25 to 28 per mil. But if you look at two, or you analyze compounds from two closely related species, it probably constrains variation in this species. So by analyzing those alkanones, we've got some of those constrained. But we also have calcification could play a factor. The factor limiting growth, either nutrient or light, or active transport, both the inorganic species utilize and that mechanism of transportation, transport. For E. huxleyi and G. Gephyrocaps oceanica, we can constrain some of those. These are the alkanone-producing haptophytes. They're both about the same cell geometry, so they limit variation in cell geometry. They probably limit variation in the isotope effect associated with Rubisco. And at least Emiliani huxleyi appears to be mainly a CO2 user. It doesn't have much carbonic anhydrase. It has a low and constant use of bicarbonate, but it is a haptophyte, so it has the potential for this light-stimulated CO2 transport mechanism. And growth rate in the oceans are limited by nutrients and light. So there have been some experiments done with Emiliani huxleyi. I showed you data from some of those, and that's both the calcifying and the naked strains of Emiliani huxleyi. These were done in Chemostat culture. This other paper here by Ulf Riebesell and his group used a batch culture experiment. So batch culture, chemostat, what's the difference? Okay, here's a diagram that shows a chemostat culture. This is a cool way to grow marine microalgae. You put the algae in here, it's a growth chamber, and there's a nutrient medium reservoir. And the nutrients in there are, you get high concentrations, maybe 100 micromolar nitrate. That's really high, but they're set at a ratio so one of those nutrients, typically nitrate, is limiting. So the algae in here, they're bathed in seawater, probably nutrient pretty similar to the concentrations of nutrients here, and they're allowed to grow and strip those nutrients. As they grow and strip those nutrients, that's basically a batch culture. You You throw algae in, you let them double until the nutrients are depleted. You can measure their rates of growth. And that's basically what the Ribosel group did, but they were smart about it, is they allowed the cells to go through only a few doublings so that there was only a small perturbation of the inorganic carbon system here, so they could measure the growth rate and the CO2 concentrations, etc. For a chemostat culture, once the nutrients are stripped, you can turn on a pump that drips this nutrient in, and so they grow only as fast as you dilute this with this water, which contains a nutrient. So if you increase the pumping rate, you increase their growth rate. You can only increase it so high because if you exceed their maximum growth rate, then you come in the morning, your nice green culture is clear. It's depressing. I've been there. So you got to back off the pump and reach a rate at which they can grow. But the key is that basically the dilution rate sets the growth rate because there's a limiting nutrient here in the nutrient reservoir. Okay, you can take samples through the overflow. You can mix gases in there so you can set the CO2 concentration. Okay, so the experimental methods are different. Each experiment tells us something. I mean, these experiments that I was involved in, we changed growth rate, we changed CO2, we can tell that both those parameters affect growth rate. Here, growth rate was measured, it wasn't controlled. CO2 concentrations were controlled, but you know, there could be just a simple extrapolation of a polynomial that fits all of these data, and that might be the applicable fractionation model, but we don't know. The key is, do any of these cultures mimic nature? They probably don't. There's no competition for nutrients, there are no grazers present, it's a non-natural system. So what the advantage is, again, of Emiliani-Huxley is that it produces those unique compounds. So we can begin to test this in the ocean, in the real world, if we can make some of these measurements. And we can. We can measure the growth rate of these organisms in nature with a little bit of imagination. Okay, so I'm going to show you data from 3 different sites. One of those sites is Station Aloha. It's a oligotrophic ocean habitat assessment. My colleague Dave Carl's really into acronyms, and this is one of his favorite. This study, it's a long-term time series study, began in November 1988, and it continues to today. There have been 294 cruises to this site to measure the same parameters that describe the physical oceanography as well as the biogeochemistry of this region. It's been highly successful because this is like the desert of the ocean, and you wouldn't expect much variability. And what they've shown is that even in this highly oligotrophic region, there's an extreme amount of variability. So this gives us one end member, an end member that is very low nutrients and has pretty stable physical conditions. And we went on 4 different cruises. This was a cruise from hell, I tell you. It was in the early, it was 2003. We started in Seattle and had a transect across the date line here and up north of the Aleutian Islands in the Bering Sea shelf, and then ended in Dutch Harbor. It was a cruise from hell because right about here, the sun went away. Have any of you been out at sea in the subarctic Pacific in the summertime? You know what? The sky is incredibly clear, but there's this sea fog that sits right above the ocean. We cruised into this sea fog, and every now and then it would open up, you'd see these little windows and realize there isn't a cloud in the sky, but it's dark because of this sea fog. It was really depressing, especially if you come from an environment like me from Hawaii where it's sun shining a lot of the time. The other neat thing, well, not so neat, it was a new ship, the Kilo Moana. It's this swath vessel. And, it was government contracts. The bridge had this console with all the instrumentation, and it had serial number 0001. Bad sign. We crossed the date line, and everything inverted in our navigation system. This nice screen where you have the ship there, and all the colors changed, and the second mate called up the company, and they said, "Oh, you didn't tell me you were going to cross the date line when you ordered this. It's an ocean-going vessel." Anyway, in essence, this is a transect that changes in nutrients. This is a high nutrient, low chlorophyll region where nitrate and phosphate concentrations in the water increase as we go from east to west. The last is one of my favorite places in all of the ocean. It's the Sea of Cortez. The fishing there is great. And we did 4 cruises, 2 summer cruises and 2 winter cruises. The early cruises focused on the Guaymas Basin, but in 2005 we went up here north of the Midriff Islands and sampled the Carmen Basin as well. And the reason that we pick these different environments is illustrated here by looking at phosphate profiles with depth. Station Aloha is a oligotroph. You can hardly measure phosphate concentrations. Dave Carl had to invent a new method to measure the concentration of phosphate that was so low. Again, the subarctic Pacific is a gradient in phosphate concentrations. We'll look at this site here, but others as well. And then Guaymas Basin has a really shallow mixed layer as well as a shallow nutricline. You can see concentrations of phosphate increase. So photosynthesis up here taking up nutrients, and they're being respired here. But you don't see the increases. The nutriclines are down over here for these other regions. So again, very different oceanographic conditions. And to do this work, you've got to get samples. The concentration of alkanones in seawater ranges from a couple nanograms per liter to about 25 or 30. So we use things like this. It's a pump filled with batteries. It operates this thing. It sucks water through a filter here, and you can filter even up to thousands of liters of water. And when you get those particles, you can isolate and measure the concentration and isotopic composition of alkanones in the suspended particles. The other thing we did is we measured the growth rate of the alkanone-producing algae by using a simple method of putting a little bit of 13C labeled bicarbonate. So we collect water from depth, put it in some jugs, add a little bit of 13C labeled bicarbonate, and then put them back out in the ocean, let them float around for 24 hours, and bring them back, filter it, and see how much of that bicarbonate was taken up into the alkanone. The rate of uptake is proportional to the rate, well, it gives us the rate of growth. The equation is here. You basically look at the activity of 13C in the compound of interest at the end of an experiment versus a natural sample, and divided by the difference between inorganic carbon added in the natural sample. You don't have to add much. We increase the concentration to about 200 per mil. Okay, here's some evidence from laboratory studies, and the slope here, this is a plot of calculated growth rate based on the 13C labeling of cells or of the alkanone versus the measured or set growth rate in culture. And these slopes are not different from one, and it shows that, yeah, we can measure the growth rate in the field of even this specific alkanone-producing haptophyte, which are 2 species in the ocean. So what we do is we have a floating array. We have a float here. There's a strobe on it so nobody runs it over at night. There's some transmitters that allow us to find it and follow it around. We had a radio RDF, radio transmitter. Look at radio waves. You get within about 6 miles of it and you can hear it ping. So we use that for a long time, but if it gets 6 miles away, you don't hear it anymore, so you kind of have to do dead reckoning. And well, we think that it was drifting this way or that way. But one day I was looking through the internet, and there are little GPS transmitters now sold to really paranoid hikers, where somebody can follow where you are by keeping one of these things on you. It transmits a signal like every 10 minutes or so, and your loved ones know where you're hiking. It's like, but I strap one of those on, and guess what? It works in the ocean, most places. There's some places where satellite coverage is not very good, but we can follow it around. We can find it. There's some of these floats here, so there's a long line between those two, because the way you get this back on is really high tech. You take a grapple and you throw it, and you grab hold of that line and you pull it in. There's some floats here, and hopefully you got your buoyancy right. These floats keep it aloft. If the floats are not strong enough, the thing goes down to the bottom of the ocean. There's a weight here, and we strap on some of these carboys. It's kind of fun work. This is the way we sample it. We use something called a rosette, a CTD rosette. It's an instrumented package, this round package. These are sample bottles. They're open at the top and bottom. They go down to a particular depth. You send a signal, and the top and bottom closes, so you get a water sample. There's also an instrument package here. There's something called a CTD, which stands for conductivity, gives us salinity, temperature, so a temperature sensor, and pressure, which gives depth. There's a fluorometer, gives us chlorophyll A concentration. There's oxygen sensors, there's light sensors, and again, these sample bottles. To do this work, you don't want to light shock any of your phytoplankton. So usually start at about 1 o'clock in the morning. So you get everybody out of bed and go, okay, we're going to begin these experiments, and you start collecting water with the CTD, bring it up on deck, and transfer it to these polycarbonate bottles. You know what the density of seawater is? A little bit greater than 1. So how much does one of these 25-liter carboys weigh? It's about 80 pounds or so. God, for the first time in my life, I developed pectoral muscles from hauling these things around. So we collect those things. We add a little bit of 13C labeled bicarbonate and close them up. We put them in these bags here. They're made out of cargo net. And then we attach them to a line here and deploy the bottles. We deploy the floats and everything else, and then pray it all comes back 24 hours later.